I'm very happy to be here telling you about some recent work we've been doing looking at material properties of the 2D Voronoi model. So this is a model of densely packed disordered cellular tissue. And what I'm really hoping to convince you of today is that this model is very weird. So in particular, what I mean by that is that the behavior of this model deviates significantly from what you'd expect of a standard glass-forming material, which at initial glance is what it looks like it describes. And what our results really highlight is that we need a better understanding of how material constituents and interactions lead to particular emergent phenomena in materials. And we also need to understand how these results are applicable when we're actually trying to use these models to describe real cellular tissues. Okay. So what is it that these models have been designed to try and study? So much of the work using these models has been inspired by this observation that these densely packed cellular tissues can undergo a transition from a fluid phase into a disordered solid phase. And this is very reminiscent of something like a glass or jamming transition that's observed in soft materials. But these densely packed cellular tissues can also do much more complex things that we'd also like to try and understand. So for example, they can do things like undergo collective cell migration, which is very important for things like cancer cell invasion, and getting sort of more complex still, these dense things that start out as simply a dense collection of cells during embryo development will undergo these complex shape changes and flows to go from being a blob of cells to something that is in fact an entire functioning organism. So at the big picture level, what it is we want to try and understand is how do you go from a single cell, just to be clear, not the same cell, but from a single cell and sort of the subcellular processes going on there up to the scale of an entire organism. So we know there's lots of different things involved. There's things like the cytoskeleton that help the cell move. Once you start getting collections of cells, things like cell-cell junctions become important, as do things like genetic patterning and stresses, etc., in allowing the different regions to differentiate, and so to get you up to an entire organism. So we're certainly a long way from understanding all of this, but studying cells sort of at this scale, so at the scale of densely packed cellular tissue, where you have these collective emergent behaviors, but maybe without the complexity of these further stages, gives us a good start point for trying to determine some of the key features of collective cell behavior and cellular interactions. And even in 2D, which is what we'll be focusing on today, this is very relevant to things like epithelial sheets, which exist throughout your body. So for example, in your skin or the lining of your gut. And so these types of cellular systems have inspired a lot of experimental work as well as theoretical and computational modeling. And in particular, what I'm going to be talking about today are these highly coarse-grained geometric models, sort of coarse-grained at the level of the cells. So just to briefly give an overview of sort of some of the main geometric models. So the one we're going to be talking about today is the Voronoi model. So in this model, the degrees of freedom are the cell centers, and then the boundaries of the cells are defined by a Voronoi tessellation of the space. So there's no information about the subcellular level. A very similar model that shares a lot of features is the vertex model. So here, as in the Voronoi model, the cells are polygons, but instead of the degrees of freedom being the centers of the cells, the degrees of freedom are instead the vertices of the polygons. So typically each cell has about 6 vertices associated with it, and each vertex belongs to 3 cells. Another class of geometric models, which is, shares a lot of features with these two, but is slightly more different, is the cellular pots model. So this is typically a lattice-based model where each lattice site has some configuration. It's a little bit like a glorified Ising model. So each site can be in one of many configurations, and sites are preferentially in the same configuration as their neighbors, and then there's some interaction energy, so that that's what happens. So, as I say, these are all highly coarse-grained models. They throw out a lot of the biology, but they have had some successes in capturing the behavior of real cells. So in particular, one good start point is that the shapes of the cells, particularly in these polygonal tilings, do actually agree fairly well with the shapes of real cells. So it's been shown that there's around 90% agreement between the shapes of real cells and a Voronoi tiling of the same cells. Another thing is that, as I said, this is an inspiration for all of these work, but these models do undergo these fluidity transitions between this disordered solid and a fluid, as is observed in real cells. But, and this is what I'm going to be telling you about today, these models have highly anomalous material properties that deviate from standard expectations. So what I'm going to tell you about today is some work we've been doing on the glassy dynamics, which are highly anomalous compared to standard glass formers. And then I'll also talk briefly about some very recent work we've been doing looking at some of the nonlinear rheology in these models. But one thing I just want to highlight at this point is that although we have these different geometric models, and they're often used largely interchangeably, we actually don't know the full extent to which these different models capture the same underlying physics, and therefore how interchangeable they actually are in making predictions about biological systems. So as I've said, today we're going to be talking about the Voronoi model in 2D. So just to give a little more detail, so in this model, the cell centers are the degrees of freedom, and as I've said, the cell boundaries are defined by a Voronoi tessellation of the space. So each cell has some area A and some perimeter P, and then we define this energy where each cell has some target area and some target perimeter. And so in our implementation of the model, all the target areas and all the target perimeters are the same. And so as you can see, if every cell can achieve its target area and its target perimeter, this minimizes the energy. So in this representation of the model, this target perimeter P0, this is also known as the cell shape index, and this is a very important parameter in characterizing the behavior of the model. So in particular, in the 2D vertex model, there is an athermal rigidity transition at some critical value of this shape index P0, which is around, well, it's at 3.81. So if the shape index is less than this critical value, the model is a disordered solid. It can't, the cells can't all achieve their target shape index, whereas above this critical value, all the cells are able to achieve their target shape index, and the model behaves like a fluid. Here we're thinking about this model more as a material. I know this is an active matter conference, but we're thinking about a thermal system. You can think of temperature like activity if that makes you feel better. But essentially, in this model, when you include temperature, you still have this solid phase and a fluid phase, and as you increase the temperature, the phase boundary between, or the P0 at which you go between your disordered solid and your fluid shifts to lower values. Okay, so as I've said, this is a model that has a disordered solid phase and a fluid phase. It looks very like a glass. So a few years ago, Daniel and Christina and co-workers set out to look at some of the glassy properties of this model. And so this is a result from their work, and what it's showing is how the relaxation time of this model varies with temperature, or inverse temperature. And so the relaxation time is essentially the timescale it takes for cells to rearrange in the system. And so as you approach the glassy regime, you expect that the cells take longer to rearrange. And so that is indeed what you see in this model, but the way that the model does this is really strange. So you see that on this plot, we have this highly sub-exponential behavior. So this is the data for the Voronoi model at different p-naughts, and it's sub-exponential. But in every other glass-forming system ever previously studied, this relation has always either been exponential, which is termed a strong glass, or faster than exponential, which is a fragile glass. So although the model does have this glassy behavior in that it shows this relaxation, the way it does it is really strange. So can we understand more about what's going on in this model and what other features may or may not be strange, given we have these unusual relaxation dynamics? So we've set out to see what else we can learn. And as a start point, this is actually just showing, if you like, that same data, but in a slightly different way. So what we're seeing now is, instead of being the x-axis being inverse temperature, it's now temperature, and we have a log-log scale. And so what we see is that as we shift this p-naught value, we go from a regime where the relaxation time increases fairly sharply over a narrow range of temperatures to a regime where we have something that looks almost like a power law relation. So for the relaxation time to increase by several orders of magnitude, there's also several orders of magnitude change in temperature. So now we can also look at the viscosity. So typically, you would expect that the viscosity follows a similar trend to the relaxation time, since they're both involved with the motion of the particles. But what we see when we look at the viscosity is that, again, at this high, at the low p-naught values, we have this nice divergence of viscosity over a fairly narrow temperature range. But as p-naught increases, we see that the viscosity seems to diverge less and less, to the point that for this highest p-naught, even though the relaxation time has increased enough that you would typically expect you would be in the glassy regime, which is often taken to be a tau alpha of around 10 to the 4, we see that the viscosity hasn't even increased by 2 orders of magnitude, and it's only made it to a value of about 10. So the viscosity is really diverging a lot less, or it's not really diverging when you would expect it to. So,
So when you say viscosity, numerically, how do you measure the viscosity in these systems?
Yeah, so we're doing a stress-controlled reverse non-equilibrium molecular dynamics protocol to get viscosity, and we're looking at the small stress scale.
So, no, meaning you impose a flow and measure the stress, or you do a stress correlation, or?
We impose a momentum flux, and we measure the resulting flow.
Flow, I see.
Yep.
I see. And there's no substrate in this, so this is a momentum conserving dynamics?
Yeah, it conserves kinetic energy and linear momentum.
Okay.
If I may ask a related question, from a perspective of migrating cells, the most relevant viscosity might well be what happens if you jam the material and force it to flow through a tube with confined boundaries. And so have you done that? And what does the viscosity that you get by that kind of Poiseuille flow agree with your definition?
So we haven't done that. So our system is fully periodic, but I do think that would be an interesting thing to look at, is how does it vary.
Thank you.
Yeah. Okay. So let's see if this model has other features that are typically observed in glasses. One relation that you can look at to sort of determine when you're approaching the glassy regime is the Stokes-Einstein Sutherland relation. So this is a relation that holds in simple fluids that says that the viscosity times the diffusivity divided by the temperature is a constant. And this is expected to break down as you approach the glassy regime because of the formation of dynamical heterogeneities. So this is that as you get into a glassy regime, you have regions where there are low motility and regions of high motility, and so you get these sort of separations of scales. And the idea is that because the viscosity is more coupled to the slow regions, while the diffusivity is coupled to the fast regions, when you get the formation of these dynamical heterogeneities as the system becomes glassy, this Stokes-Einstein relation breaks down. But when we look at what goes on in our system, we don't really observe this. So what we see is that over this whole range of temperatures, most of our results follow this nice straight line. So that suggests that the Stokes-Einstein relation is not violated. And actually this data here, this the one that looks like it's a bit off, is the one that's actually doing what you would expect, in that at high temperatures, Stokes-Einstein is obeyed, and then below some temperature, it starts to deviate. And so what this result suggests is that since we don't have this Stokes-Einstein violation, this suggests that the dynamical heterogeneities in the system seem to be suppressed. So we can look more at the dynamical heterogeneities, and so what I'm showing here is a visual representation of them. So these are snapshots of our system where we're showing each cell, and then each cell is colored according to how far it's moved after a timescale tau alpha has passed. And this is showing how far the cells have moved relative to their neighbors at the initial timescale. So on the left, we see what you would expect if you have dynamical heterogeneities. So we have these blue regions where things haven't really moved much, and then we have these white regions of cells that have moved relative to their neighbors. But on the right here, at the higher P0, we have something that looks much more like static, with no clear separation of these different scales. And so we have also quantified the dynamical heterogeneities, and you do indeed see that as you vary P0, the dynamical heterogeneities become more and more suppressed. And actually, one thing that our results really highlight is that we don't actually have a very good understanding of what it is that these different dynamical heterogeneity measures are even measuring. So sort of our results and some other results from models with strange dynamical heterogeneities show quite different answers depending on which measure you use. So even though you think of these as standard measures, we actually don't really know what a lot of these measures are actually measuring in the model. But for now, in my last 10 minutes or so, I want to briefly go on to talk about some of the nonlinear rheology of this model. So everything we've been looking at so far was in the low stress regime, but as we saw in the beginning, there are actually these biological processes where large stresses are important, and that can be in things like in organ development, but even when you do something like bend your arm, you're actually putting tissue in your arm under high stresses. So just briefly, as I've already mentioned, we're doing a stress-controlled rheology protocol. So we're putting the system under stress and measuring the flow. Now, if when you do this, as you change the stress, the viscosity, the strain rate, the stress is proportional to the strain rate, that proportionality constant is the viscosity. But if when you change your applied stress, the measured viscosity changes, then you're in a nonlinear rheology regime, and this can change in a few different ways. So if, as you increase the stress, the viscosity decreases, that's shear thinning. If, as you increase the stress, the viscosity increases, that's shear thickening. And this can either be continuous shear thickening, so as the name suggests, the viscosity increases continuously with stress, or it can be a discontinuous shear thickening, where at some applied stress, the viscosity will actually jump discontinuously. Now, in this stress control protocol, you can actually get another regime where if you put the system under enough stress, it will actually stop flowing and it will jam. And so this is called shear jamming. And I guess, spoiler alert, we see all of these different things in this model. So first looking at the low P0 regime, what we see if we're looking at the viscosity versus stress is that at high temperatures we have this fairly Newtonian flow, so the viscosity is constant with stress, while at low temperatures the data here is missing because we have a yield stress, so you apply the stress, but it's not enough to make the system flow. And then once the system yields, we observe this pronounced shear thinning behavior. And so this is quite reminiscent of what's often observed in glass formers. Now, at the other extreme, at the highest P0, we see a lot of the opposite behavior. So we still have a Newtonian flow regime, and at small stresses, we get Newtonian flow across the range of temperatures. But then we see that as we increase the stress, the viscosity starts to increase. But this representation doesn't really, or this representation of the data doesn't really show us what type of shear thickening it is we have going on. So for that, we need to look at the flow curves. So what I'm showing now is the stress versus the strain rate. It's a little confusing in this representation because this is the thing we're varying and this is the thing we're measuring, but bear with me. So now we have, this is our Newtonian regime, and then over here, this is our region of shear thickening. So we have this continuous shear thickening here where we see that the curves start to deviate from the initial gradient. And then these vertical lines here, this indicates our discontinuous shear thickening. And then where you see that these flow curves are starting to go backwards on themselves, this is showing that the system is headed towards this shear jamming regime, so where the more you stress it, the slower it flows. And then in this region here, again, this data is missing. It's not because we didn't try, it's because this is the regime where the system seems to be jammed, and so you don't get a flow even though you're putting it under high stress. And I just want to briefly compare this to some recent work that came out of Max B's group looking at shearing the vertex model under a strain rate controlled protocol. So here you again see this pronounced discontinuous shear thickening, but in this type of protocol you can't see the shear jamming behavior. So this is the two extremes that we've already looked at. So what goes on in between? So in between, what we see is that as we vary P0, we gradually see this continuous transition from this shear thinning behavior into this shear thickening regime. And this transition seems to occur right around this critical value of P0 of around 3.81. So we can summarize this data a little more into some flow phase diagrams. So now I'm showing the applied stress versus temperature for each P0. And although the temperatures here are different, they are over comparable ranges of the relaxation time, so you can directly compare these plots. And so what we see is that as we increase P0, we have this region of shear thinning in the low temperature, high stress regime that gradually disappears and is replaced by this incoming region of shear thickening behavior that starts as continuous shear thickening, and then the discontinuous shear thickening and the shear jamming behavior start to appear. So we wanted to try and see if we can condense this information down more. So is there any sort of universal behavior in these different regimes? And so this is our current best attempt at a scaling collapse for this data. So this is just phenomenological. There's not currently a theory behind this. But what we find is that we can get a pretty good scaling collapse of both the shear thinning and the shear thickening regimes. And so the collapse forms are not exactly the same in each regime, but they're of similar forms. So we have dependence on the temperature, we have dependence on how far the value of P0 is from this critical value, and then we have terms that depend on the applied stress, and then we have these fit parameters, which are sigma thin and sigma thick, that essentially are the stress scales where the nonlinear rheology starts or stops. And so with this, like I say, we have a reasonably good scaling collapse across these two different regimes. But one thing I should note is that these scaling forms are quite different to recent studies showing universal scaling collapse in glassy materials on the shear thinning side and in dense suspensions on the thickening side. And we've tried forms more along those lines, but they don't give as good of a collapse as this does. Okay, and so with that, I will leave the longer summary, but hopefully I've convinced you that these models are quite weird. They have very unusual material properties that we need to understand, both for our understanding of materials, but also in the applicability of these models for understanding real cellular systems. And so with that, I'd like to thank Daniel, who I did this work with, and Chengling, one of the grad students in our group who did some of the work on the first part. I'd like to thank the funding, and thank you all for listening.
We have time for questions. Christina.
Yeah, I was wondering, going back to the questions about the linear shear viscosity, whether you also try to calculate it from, say, a Green-Kubo formula as a time integral of stress correlation function. Because a few years ago, we did that for actually the self-propelled Voronoi model, where it's kind of not justified, but we did find it to diverge in that case. So it was not just curious.
So I think when we were very much starting out, sort of testing this algorithm for doing the viscosity, Daniel did do a couple of sanity checks of are the numbers coming out comparable with Green-Kubo, but we haven't done it extensively in that way because it gets difficult. So yeah, we haven't done it across the board, but we did do a few checks at the beginning.
All right, please.
Great talk. Great talk. Thanks. So I was wondering, first of all, the temperature you calculated by the mean square displacement, right? In your case, in the self-propelled Voronoi, what is the temperature for you?
Oh, so we're simulating a thermal system, so we're imposing a Nosy-Hoover thermostat. So we put the temperature in.
You put it in. So it's not like the mean square displacement. It's not. You don't calculate it from that.
No, it's a thing you put in.
Ah, interesting. Okay, cool. And I have like a second question, if that's okay. Is that okay?
Yeah.
Sure.
Thank you. So, like, you saw us in the first couple of slides. You had this Pika-Chamara picture with a regime of exotic whatever in between the solid and the. Did you see any differences in the viscosity in the solid, exotic phase, or is it exactly the same? Or
yeah. So one thing I didn't say is that in these plots so the solid markers are indicating the regime where the model is fluid, and then the open markers here correspond to regions where you have hexatic ordering. So the divergence very much starts around when the hexatic ordering kicks in.
Please.
Yeah, so I'm quite curious about the dynamically homogeneous glassy phase. So I was curious what the 4-point susceptibility would look like. Is it just flat overall and has very small values, or are there any non-trivial signatures at other time scales other than your typical alpha relaxation times?
Yeah, I think I have that on the slide here. Okay, so this is the peak height of Chi4 for different tau alphas. So typically you would expect that this would vary by sort of at least 2 orders of magnitude over this range of tau alphas. So yeah, we see there is some increase in the peak height of Chi4, particularly at the lowest P0, but again, like at the highest P0, this isn't even changing by 1 order of magnitude.
Interesting. Okay, thanks.
Did you have a question too? No. Let's go ahead then.
Yeah, I'm curious, now that you have these models and you're looking at shear thickening, shear thinning, maybe you mentioned this, but can you look at what has happened statistically with forces inside more precisely to understand where these phenomena come from? since you have the whole simulation and everything.
We definitely can. That is a thing that we would like to do, but have not.
Maybe statistics of force chains.
Yeah. We're definitely thinking along those lines. Okay. This is very recent, and we just haven't quite got to that point yet.
Great, thanks.
Yeah.
Other questions?
Can you go back to collapse slide?
Yep.
So, like, I know you said you don't have a theory for it, but do you have like an intuition why the scaling of the parameters is different in the two regimes?
Not really. Honestly, no. It's I would love to have an actual theory or intuition as to why these forms work, and in particular why I could get similar form, like quite similar, but not not the same, or not the same, but just like flipping one sign or something type forms to work. So yes, I would like to do that, but I do not yet. But if anyone has any genius ideas, let me know. Yeah.
More questions? Do we still have some time? Actually, I have one myself. I guess it's related to Christiane's question, but how do you interpret the lack of class dynamics at high P naughts? Is there some understanding that you have of why it doesn't emerge?
As to why it doesn't, I don't think we have a strong understanding of why. So there are other glassy models that show this similar behavior with this lack of dynamic heterogeneity, and several of those are also involving sort of many body interactions, and sort of a lot of the work is actually inspired by trying to approach the mean field limit from 3D. So I would suspect it's related to the fact that this isn't just like a pairwise interaction model, but beyond that, exactly what is going on that's making this happen, again, we don't know.
Thanks. Is there any final question then? All right, if not, I think we can stop here for the coffee break. Thank Helen again, and we'll